Imagine I gave scientists a tiny sample of your blood, but I removed your name, your birthday, and every detail about you. Could they still estimate your age? Surprisingly, yes. But here's the strange part, they are not reading your DNA sequence like a birthday certificate. Your DNA is something you inherit from your parents. It's the same genetic instruction book you have carried since before you were born. Apart from a few rare changes, that DNA sequence stays almost the same throughout your life. So, how could it possibly reveal whether you're 25 or 65? The answer lies not in the DNA sequence itself, but in tiny chemical marks attached to our DNA. These marks are called DNA methylation. You can think of them like tiny sticky notes attached to different parts of your DNA. They don't change the DNA sequence itself. Instead, they are tiny chemical marks that add another layer of information on top of our DNA. Unlike our DNA sequence, these chemical marks are remarkably dynamic. They can change throughout our lives in response to normal development, aging, and even aspects of our environment and lifestyle. And here's the fascinating part. As we grow older, some of these DNA methylation patterns change in surprisingly predictable ways. So predictable, in fact, that scientists have used them to build what are known as epigenetic clocks, tools that estimate a person's age by analyzing patterns of DNA methylation. But how can tiny chemical marks on our DNA become such accurate indicators of age? That's exactly what we'll explore in this video. But before we can understand how these clocks work, there's one more important idea. Our bodies aren't made up of just one type of cell. Depending on the organ or tissue, we have many different kinds of cells. We have brain cells, blood cells, liver cells, skin cells, muscle cells, and many more. Although these cells perform very different jobs, they all contain almost exactly the same DNA sequence. So, what makes a brain cell behave differently from a liver cell even though they contain exactly the same DNA? One important reason is that each cell type has its own unique pattern of DNA methylation, which helps determine how that cell functions. Think back to the tiny sticky notes we talked about earlier. Every cell has the same instruction book, that is, almost exactly the same DNA sequence. But the sticky notes are placed in different locations throughout that book depending on the type of cell. As a result, every cell type has its own unique DNA methylation pattern. For example, the pattern found in a blood cell is different from the pattern found in a brain cell, a liver cell, or a skin cell. Scientists call this tissue-specific DNA methylation. But there's one more piece of the puzzle. These tiny sticky notes or chemical marks aren't attached randomly throughout our DNA. Instead, they appear at specific locations. You can imagine our DNA as an enormous instruction book with millions of possible places where these tiny sticky notes could be attached. As we grow older, some of these locations gradually gain DNA methylation while others gradually lose it. Individually, these changes may seem small, but when researchers examine thousands of these locations together, they find that many of these locations change with age in surprisingly consistent ways. This raised two important questions. First, if these tiny chemical marks found at specific locations in our DNA changed with age in such predictable ways, could they actually be used to estimate a person's age? And secondly, there was another challenge. We just learned that every tissue has its own unique DNA methylation pattern. So, would scientists need a different clock for blood, another for brain, another for liver, and another for skin? Or could one single clock somehow work across all of them? In 2013, researcher Steve Horvath set out to answer exactly these questions. To do that, he assembled one of the largest collections of DNA methylation data available at the time. His study included nearly 8,000 non-cancer samples from 51 different human tissues and cell types, including blood, brain, liver, kidney, lung, skin, saliva, and many others. So Steve Horvath wondered, could he identify a small number of these DNA methylation locations that changed with age so consistently that they could estimate a person's age regardless of which tissue the DNA came from? After analyzing more than 21,000 DNA methylation locations, the answer was yes. His model identified just 353 DNA methylation locations that, when analyzed together, could accurately estimate a person's age from many different tissues and cell types. Together, these 353 DNA methylation locations became the foundation for what is now known as the Horvath epigenetic clock. But did it actually work? Could it really estimate the age of people it had never seen before? Yes. To find out, Horvath tested the clock on completely independent DNA samples that had not been used to build the model. And the results were remarkable. The estimated age and the person's actual age showed an extremely strong relationship with a correlation of 96%. Even more impressively, for about half of all the samples, the estimated age was within about 3.6 years of the person's real age. In other words, if someone was 50 years old, the clock might estimate their age as 47 or 53 rather than 20 or 80. For something based on just 353 DNA methylation locations, that's remarkably accurate. So, does this mean Horvath's clock was measuring biological age? Well, not exactly. Horvath's clock was originally designed to estimate a person's chronological age, that is, how many years they had been alive. And it did that remarkably well. But researchers soon also noticed something interesting. Sometimes the clock estimated people to be older than their actual age and sometimes younger. Those differences often turned out to be biologically meaningful. This suggested that DNA methylation might be telling us something more than just the passage of time. It might also be capturing how differently people of the same age are actually aging. Think about two people who are both 60 years old. One exercises regularly, has healthy blood pressure, healthy blood sugar, and no major illnesses. The other has diabetes, heart disease, and struggles with everyday physical activities. Chronologically, they are exactly the same age, but biologically, they may be aging quite differently. The challenge was this. Because Horvath's clock has been trained to predict chronological age as accurately as possible, it mainly learned the DNA methylation locations that best tracked the passage of time. But researchers also realized that some DNA methylation locations don't just reflect the passage of time. They may also capture whether someone's body is aging faster or slower than expected for their age. Those biologically informative locations could easily be overlooked when the goal is to simply predict someone's birthday. That realization inspired the next generation of epigenetic clocks. In 2018, researcher Morgan Levine and her colleagues took the next step. Instead of training DNA methylation to predict a person's chronological age, they first asked a different question. How can we estimate a person's biological age? To answer that, they turned to health data from nearly 10,000 adults. Rather than looking only at how old people were, they examined a wide range of routine clinical measurements. Things like blood sugar, markers of inflammation, liver function, kidney function, immune system measurements, and several other indicators of health. Then they asked which combination of these measurements best predicted a person's long-term risk of death. The result was a new measure called phenotypic age. Unlike chronological age, which simply counts how many years you have been alive, phenotypic age estimates how old your body appears to be based on how well multiple biological systems are functioning. Think back to our two 60-year-olds. Based on these health measurements, one person's body weight resembles that of a typical 50-year-old, while the others might resemble someone closer to 70. Their birthdays are exactly the same, but their phenotypic age is different. And that difference may reflect how quickly or how slowly their bodies are aging. Now came the clever part. Once the researchers had created this estimate of biological aging, they asked a new question. Could DNA methylation predict phenotypic age? In other words, could patterns of DNA methylation estimate not just how much time has passed, but how biologically old a person's body appears to be? To answer that, they used DNA methylation data collected from another group of participants and searched for the DNA methylation locations that best predicted phenotypic age. After analyzing more than 20,000 DNA methylation locations, their final model identified 513 DNA methylation locations. Together, these formed a new epigenetic clock called DNA methylation PhenoAge. But did it actually work? Could this new DNA methylation clock really capture biological aging better than the earlier clocks? The answer was yes. The researchers then validated the DNA methylation PhenoAge across 5 large independent population cohorts. They found that people whose DNA methylation PhenoAge was higher than expected for their chronological age were more likely to die earlier. They were more likely to have multiple coexisting diseases and even more likely to experience poorer physical functioning and also less likely to remain disease free. When they directly compared it with the earlier Horvath and Hannum clocks, DNA methylation PhenoAge predicted both 10-year and 20-year mortality more accurately. In other words, this new clock wasn't just estimating how much time had passed, it was capturing meaningful differences in biological aging between people of the same chronological age. Today, epigenetic clocks continue to evolve. Scientists have developed many different clocks, each designed to answer a slightly different question. Some, like Horvath's clock, were designed to estimate chronological age. Others, like DNA methylation PhenoAge, aim to capture biological aging more accurately. More recent clocks have gone even further. Some are designed to predict lifespan, others estimate the pace at which a person's body is aging. And researchers continue to improve these tools every year. Although no clock is perfect, together they have transformed the way scientists think about aging. Today, researchers no longer see aging as something measured only by the number of birthdays we have celebrated. Increasingly, they are learning to measure it through biology itself.